So guys, we're about ready to get started after a little bit of a hiccup while we got set up. Our speaker today showed up with this very unusual operating system called Linux, which gave us a little bit of a hiccup here, but I think everything is working. So let me introduce our speaker. Jeffrey Mulligan is a neighbor of ours here at Carnegie Mellon. He's in the Human Systems Integration Division at NASA Ames next door. And his work is focused on the development of video-based eye tracking systems and the application of these to applied problems such as reading from by operator state assessment and interface design, which I think we'll learn more about. I'd just like to mention, though, that tracking eye movements is tremendously useful and interesting because it's very, very revealing of the cognitive processes people are going through, where and what they're paying attention to in very small movements of time, which tend to map very, very closely to cognitive architectures and the way people pay attention, what they're thinking about, and so on. Much of that information is below the conscious level, and those techniques have been used in analyzing how people pay chess, what they pay attention to in interfaces, and in many, many other areas. So it's really a very powerful tool, and I think we'll learn a lot about it today.
Well, thank you for that introduction, and I have to say that I think, unfortunately, although the cognitive stuff is an interest of mine, that's not what I'll be talking about today. So, but hopefully you learned something interesting anyway. And so basically the talk today is about how we can use eye movements to learn something about fairly low-level visual processing. And so my collaborator on this talk is Scott Stevenson from the University of Houston. And you'll forgive me that the remote people aren't going to be able to see any of the live demos, but I have to switch 2 slides every time. So Scott and I have been working together on this for a little over 10 years, and his lab is very well equipped with a very sensitive instrument for measuring small eye movements. So this cartoon kind of illustrates the problem. The caption is 4-wheel drive, and you notice that all the people in the car, they each have a steering wheel. And this is kind of the problem we face when trying to study eye movements, that there's a lot of different mechanisms in different parts of the brain that all are tugging on the wheel, trying to make the eye go one way or another. And so what we'd like to do is to, learn the characteristics of each one of those, but all that we can observe is the motion of the car or the eye. And so some of the different things are listed over there on the left. We have fixational eye movements where you try to keep your eye trained on one thing. Saccades are very rapid ballistic movements that go from one fixation point to another. And smooth pursuit is sort of like fixation on a moving object, where something's moving and you want to maintain your eye on that object. VOR is the Vestibulo-Ocular Reflex. That can be easily illustrated if you hold your finger up in front of your face and move your head back and forth. You can pretty easily keep your eye trained on your finger. If you move your finger at the same speed, you can't. And the reason is that when you move your head, you have a very fast control loop from your balance organs straight to your eyes that can cause fast stabilization movements, whereas even though you're voluntarily moving your thumb, that voluntary signal can't be copied to your eye to move your eye to go with your finger. You have to use the visual input. You have to use the image of your finger to then control the thing, and that basically cuts out above 2 hertz. So 2 main classes of eye movements are version and vergence. Version is when the 2 eyes move together, which is mostly what we do when you're looking around. Vergence is when they move in opposite directions. And so horizontal vergence is commonly used when we look at things at different depths. and you can do that voluntarily. Vertical vergence, there's a small amount necessary basically for looking at things that are different depths that are off in oblique directions, but generally these are not large and they cannot be done voluntarily. If you put a prism in front of one eye that deviates the line of sight up or down, then the eyes will correct for that, but that's done reflexively and you're not really aware of it. Anyway, that's the game, is that we're going to try to use eye movements. We're going to present different things, different stimuli to the visual system, and then see how the eye moves differently. And the general philosophy is that the back end of the motor system is just doing the same thing all the time, and so that any differences we see will reveal differences in the front end processing. And so I'm going to show a demo here that the remote people won't be able to see. I apologize for that. What you see here is there's 3 spots moving from side to side. And this has been designed so that the center dot is supposed to be more or less the same luminance as the background. And what you're supposed to try to notice is whether all 3 dots move together. And when the center one has the luminance which is exactly matched, it will sometimes seem to lag between the other 2. So I see a few people nodding. I can click on this and, oops, I guess I didn't want to click on that. Well, anyway, oh, there we go. I think, yeah, so this is controlling now I'm changing the luminances, and so for me, right about there is where the middle one is neutral, maybe a little bit dark. But that varies a small amount from person to person, so that's the reason we have it adjustable. But the basic effect that I'm trying to demonstrate is that the target defined by an equiluminant color is processed more slowly, and it appears to lag behind the other targets. And so then the question is, if you actually see it lagging, will your eye lag behind? Will there be a longer temporal latency when the eye tries to follow that target? And if so, then we can use that as a way to investigate lots of visual processing. So I'm going to go ahead and give you give the punchline of this talk first. And so then if we get lost in some of the technicalities, you don't have to feel bad about missing it. Basically, I'm going to be talking mostly about smooth eye movements. So I'm not talking about saccades or fixation, but when the eye smoothly follows a moving target. And traditionally, there have been 2 different types of smooth movements that have been studied, and historically, they've sort of been defined on the basis of what the stimulus is. So smooth pursuit is when you use a small point target and you say, okay, follow this target. Whereas if you have a large field and you move that field, then people's eyes tend to move in response to that, whether they're trying or not, and it's not a small target. And they call that ocular following. And it's usually done with a large field and involves a lot of peripheral vision. So our view, which has been developed over some time, is that there's 2 systems, voluntary and reflexive, but that both of these can be engaged by the 2 types of targets. So if we have a small punctate target, that yes, you follow that voluntarily, but that the reflexive mechanisms are still driven as well by that. And similarly, with with a large full field target, you can voluntarily try to follow that or not, and you have the reflex going on at the same time. In the ocular following case, it's more sort of biased towards stimulating reflexes. But we think that the distinction into pursuit and ocular following is really an artificial one, which is based on the stimuli and not on the effects. And so what we're going to see is that there's a couple of sources of evidence for this, and one of the important ones is that there are reflex-only movements, most importantly vertical vergence, that you cannot do voluntarily, and that there are certain types of stimuli which aren't capable of evoking those movements. And similarly, we have an experiment with multiple targets where you have to select one with attention, and then we see different time course for we see reflexive responses to the unattended stimuli, and they have different temporal dynamics. So now we'll just get into the details. So I'm going to start with a figure from a somewhat old review paper on smooth pursuit from a lab up in San Francisco. And so this figure up above shows the what's the basic control loop. And is this a pointer? I guess nobody knows. Oh, there we go. So we have the target hitting the retina, and then there's motion of the target on the retina. That goes up to the brain and gets decoded. And then eventually there's a pursuit command, which is sent to the motor system, and the eye moves in response. And then this having a plus sign and this a minus sign means that the effect of the eye motion is to cancel out the motion of the target on the retina and stabilize it. So the whole goal of this this feedback loop is to keep this signal here 0. And I'm not going to talk about the bottom panel. So this is a slightly more complex version from their paper, and what they've added here, they have a 90 millisecond delay, and then they have these 3 mechanisms. And RAE stands for retinal acceleration error, RVE is retinal velocity error, and RPE is retinal position error. So they compute these 3 different errors and run them through different nonlinearities and add them all together and then integrate the signal and get a motor command. So they came up with the details of this from experiments on monkeys, and so the main difference that we're proposing here is that we think that this delay is not a fixed 90 millisecond delay, but depends on what's happening in visual processing, that there are different types of stimuli take different amounts of time to be processed, and that, again, that we assume that all the rest of this is sort of a constant system, but that by looking at what happens when we change stimulus properties, we're learning something about this early stage. And so now we get to the part that the remote people are going to have to imagine, because they can't see what's on the screen here. This is the task that we used. We just have a stimulus that jumps around randomly on a random walk, and you're supposed to keep your eye on it as best that you can. And those of you in the audience here, you might try it and see how easy or difficult you feel that it is, but most people don't complain too much. So, what we're going to see next is a, oh, I guess I have to. Now we've got the same thing. The green spot is still the target, but the red spot indicates the line of sight of an observer who's trying to do the task. And what we can see is that he's not able to do it perfectly. The red dot kind of lags behind the green one. It doesn't move quite as quickly, and it has these little jumps. So those jumps are the saccadic eye movements. But we're interested in the smooth component, and you may notice that that in between the jumps it seems to kind of vibrate around. That looks like it's not really doing anything to get it towards the target, but as we'll see, it actually is. Those little vibrations are movements that that are driven by the motion of the target. And this next demo is going to take a few seconds to load. This laptop's a bit slow, but it's showing just the same thing we saw, except that there will be 3 replications from the same subject and 3 replications from a second subject. And so the 3 red dots are me and the 3 purple dots are Scott. And the main point here to notice is that most of the time we're both doing about the same thing. Occasionally the 3 red dots will kind of clump in a different spot than the 3 purple dots. And so that's showing that there is some idiosyncratic difference between the 2 of us that makes the behavior be different in a repeatable way. Again, this laptop is a bit slow. It takes a long time to load the next trial, but we'll look at one more. So one thing we like to do that we're not too far along at is actually to have a detailed model of how this task is done that would be able to characterize those individual differences. So now we're going to see a demo of how we actually do the analysis. And so at the upper left, we have the raw data. The green is the position of the target, and these are horizontal and vertical traces. I think it says the upper one's horizontal, lower one is vertical. And so this is, it's a 4-second trial. And you can see that the white trace is the eye, the little jumps, those are the saccades. So the first thing we do is to differentiate both signals to get a velocity plot. And so now, again, the green and white are the target and the eye, and the little spikes, those are the high velocities of the saccadic movements. And then these white lines that are above and below the traces, that's a velocity threshold for our saccade detector. So every time one of these crosses that line, then we get a pulse down here that says, oh, a saccade occurred. And we select the smooth velocities by inverting these pulses and widening them up a little bit. And we multiply this times the velocity signals. That gets rid of the spikes. And up here, we've interpolated the places where we've cut the spikes out, and we've also done a little bit of smoothing. So when we load the next trial, you'll see that when this panel first appears, there's a lot of very high frequency stuff that gets filtered out. So now we have the target velocity in green, the target velocity in green, and the desaccaded smoothed eye velocity in white. And then finally, we complete the cross correlation of those 2, and that is shown here. And so this trial was 4 seconds. This is plus and minus 4 seconds with the green line indicating 0. And so there's a fair amount of noise, but we can see that this positive peak here to the right is saying that, well, after that small delay from 0, then if the target moves to the right, then a little bit later the eye will move to the right. And so then what happens is we repeat that. This is just another trial. There we had the velocity filtered, a new cross correlation, and then down here at the lower right, this is the average of this with what we got from the previous trial, and we do it again. And so now this is going to free run. So these are 4-second trials. We do 50 of them. It takes about 3.5 minutes. And what you'll see if you watch this lower right-hand panel is that gradually the noise out far from zero will average out and go to zero, and we're left with these little things that look like impulse responses happening just after zero. And so anyway, that's the method, and I'm going to go ahead and stop that, but we'll go ahead and look at what we get when it's all done. So this is a plot just of that the first 600 milliseconds, and in this case the white is the signal, and the green curve is a descriptive curve fit that we just used to get estimates for some of the parameters. But the features here that you can see are that we start at zero, we come up to a peak at here around 120 milliseconds. There's this curious negative dip at 200 milliseconds, which, if you think about it, that means that, well, the target moves to the right, 100 milliseconds later the eye moves to the right, and another 100 milliseconds after that the eye's moving to the left. Why would it do that? And then it bounces back to the right again and eventually settles down and goes to zero. And so here's the same thing but for vertical, and we can see that that negative dip is absent. We still have kind of some ringing here, but it's sort of riding on this long shoulder. And we get similar results for the second subject. Again, for the horizontal, we get the overshoot, and on the vertical we get some ringing, but no negative overshoot. So the way that we study this in Scott's lab, he has this machine which is the dual Purkinje image eye tracker. The Purkinje images are the reflections that come off of the refracting surfaces of the eye, and there's 4 of them. The front of the cornea, where we normally see a highlight, is the first. But you have the lens inside your eye that generates the third and the fourth. The second one is from the back of the cornea. It's very difficult to see. It's right in the same location as the first. But this instrument uses optical servos to track the first and the fourth Purkinje images, and it's very sensitive. It also has an accessory called the stimulus deflector, where you look into the machine and there's some infrared beam splitters that the tracker uses. Then you look through these little telescopes that contain mirrors that be controlled by little motors that allow you to move around what they impose movements on what you see. And that was originally done so that people could basically cancel out eye movements using the instrument. And it was discovered that when you do that, if you can stabilize things on the retina, they tend to disappear after a very short time, kind of like after images. But so we use the stimulus deflectors to generate the motions of the targets. And that is very convenient because we can basically look through the instrument and put up whatever we want on a screen, and it can be stationary, and then we generate the movements by sending these signals to the little motors. And it's a binocular instrument, so we can do this independently for the two eyes. So we're basically doing a random walk in 4 dimensions because each eye is going independently. And so I'm going to I have a few more demos that the remote people are going to miss again, but we have a few different stimuli that we used. This texture here, you sort of call it the melting texture, and the main point of the melting is that we don't really want to have a stationary feature that you can lock onto and track. I'm not sure how effective this is because the texture is still visible for half a second or something, but there's no real landmark that you can permanently lock onto. And another stimulus that we've used is what we call dynamic noise. And this is like a new noise frame every field, every frame of the movie. And so that you really, if we were to take one of the frames of the movie and move it, you wouldn't really notice because every frame is the same. I mean, it's noise. And so what's interesting is that when you move this through using the mirrors, it doesn't really generate any left-right motion signal, but it does generate a vergence, a stimulus for vergence, because the two eyes are seeing the same, at each instant they're seeing the same random noise. And then when one moves relative to the other, the eyes will still try to converge to keep that going. And when you look at it, you know, it looks like the thing is sort of moving in and out in depth, and your eyes try to follow that. So again, with apologies to the remote people that can't see the results. These are correlograms computed as we saw on the previous panel, and so the magenta here is the single spot. We get a nice big response. For the melting texture, we get a comparable response. And then for the dynamic noise, we get almost nothing. And this is for horizontal version. So this is right and left eye doing the same thing, left to right. And again, as we expect, the dynamic noise doesn't really do anything. And this is the same for vertical. And as we saw before, the vertical motions don't have this negative overshoot on the response, but similar latencies, a little over 100 milliseconds, and no response to the dynamic noise. So here we have, by contrast, the vergence response. And first of all, the response to the spot and the texture, we can see they're delayed compared to the version by maybe 50 to 100 milliseconds. And that we get a nice strong response to the dynamic noise, but that it's even slower. And the term for this, some of you may have seen the so-called magic eye stereograms, where you have to make your eyes go apart and you see this kind of figure pop out. So that's sometimes called cyclopean perception, because you don't see it until after the inputs from the two eyes have been combined. And this delay, 50 to 100 milliseconds between the yellow curve and the red one, represents the time required to do that binocular fusion, because you can't make the response to the dynamic noise until the two eyes have been fused. Whereas for the left-right version, you can, you get the signal with one eye closed. And then finally, here's the response for vertical vergence. And what we see is that the signals are much weaker, but that they are present. Go ahead.
When you were showing that dynamic noise image, it looked to me like there were horizontal lines in the image. What's up with that?
That could be an artifact of the projector, or the you know, the screen. This is not what we use for the experimental stimulus. So the screen updates are not necessarily synchronized with the display refreshes. And it could also be. Are you talking about like things at this spacing?
Yeah.
Yeah, I see those too. I don't know what they are. It's possible there could be there could be an artifact in the noise, that there's some statistical non-uniformity. I don't know. But we don't see anything like that in the real stimulus. That's just sort of for illustrative purposes. So what this graph shows, what happens when we reduce the contrast of the texture, and basically as we this is high contrast over here at the right, and as we make it weaker and weaker, we get a bit of slowing of the response. So again, that's I showed the demo at the beginning of how the equiluminant target appeared to lag behind the other one. Well, some of you may have heard of the Pulfric effect, where if you put a filter over one eye, that's actually not quite the same as this, because here we're keeping luminance constant, reducing texture, but it's another way of making visual processing slow down. And so we're seeing that slowing here, and pretty much the same for if vertical version is slightly slower than horizontal version. That difference is sort of maintained as we reduce contrast. There's sort of an contrast, sort of an additive effect. So now we're going to look at something that we call a second order stimulus. So what you can see here is there are these bullseye rings that are filled with texture, and they alternate with rings that have no texture. Or we're basically modulating the texture contrast between the rings. And this sequence is cycling through two different sweeps. In this case, well, now we're reducing the texture contrast. So we begin with high contrast in the rings and zero contrast in between the rings. Now we're doing another sweep where we add luminance. Okay, now we're doing the contrast sweep, and then we're going to switch. Now this is to the luminance sweep. So here the texture is bright, then it gets darker. And so that light dark is what we call first order contrast, luminance contrast, whereas the texture contrast that we're sweeping now is what we call a second order stimulus. And this is interesting because the models of elementary motion detectors are sensitive to the movement of light and dark. But so they are basically blind to the pattern of high and low contrast texture. You can move that modulator without moving the underlying carrier, and that's what we call a second order stimulus. This doesn't actually show the motion, but what you can imagine doing is, well, this is a dynamic noise carrier, so if we move the whole thing, it doesn't really matter, because the carrier, you can't tell if it's moved or not. But the modulator, you can see the motion of. And so what's shown here are the correlograms for 5 different stimuli. Now this C value represents added luminance contrast. So in that demo, we were adding large amounts of luminance contrast. Here we're adding very small amounts. And 2.5% is a small amount, but it turns out it's plenty to see and to make your eyes move. But what you can see is that, like the green and the yellow curves, they've lost their little overshoot wiggle, and they've been pushed out in delay by maybe 100 milliseconds. And so that's 0 is the yellow one, and minus 1% is the green one. Well, it turns out that because of well, these are difficult experiments to do because you have to calibrate your system very carefully and worry about nonlinearities in the monitor. But when you've done that very carefully, what you discover is that the perfect null doesn't happen at 0. It happens when you actually have to mix in a little bit, and it's because of nonlinearity in your visual processing that you actually have to add a little bit of luminance to the high contrast texture in order to get the perfect null. And so that's vertical and horizontal vergence. Vertical vergence, there's not much really happening here at all. But so now we've taken those results and we've summarized them by looking at the latency of the peak of the correlogram plotted as a function of added luminance contrast. And what you can see is that we start out here at like 100 milliseconds where we have typically seen things. And as we get near 0, it goes up pretty significantly. And that this peak here is, you know, close to 0, but very slightly to the right. And that the vertical vergence pretty much goes off scale, and the signal is so weak that we can't estimate anything sensibly. And second subject, similar results. And third subject. So these are similar results now, but we varied the texture contrast at the null. And again, these are pretty flat, but they go up at the very low contrast. And this yellow curve, the vertical vergence, these points are garbage. There's basically no signal, and we're fitting noise here. And another subject, and third subject. Oops. Fourth subject. So anyway, that's the first part of the story, and the main conclusion that we want to draw from that last experiment is that the second order stimulus doesn't drive vertical vergence at all. So now we're going to look at the dual target experiment, and in this demo, you'll see that we have 2 initially concentric rings that go on independent random walks. And your instructions are to keep your gaze trained on the center of one or the other. And so you might just try that to see how easy you find it. It's not too bad. Does everybody feel like they can do it? So now we're going to look at the results, and these are a little dark, but so these are, again, the correlograms, and we're looking at vertical vergence first here, and we've got two conditions. On the left is where we attend to the inner ring, on the right attend to the outer ring, and the red curve is the response to the inner ring, and the green curve is the response to the outer ring. And what we can see is that we always get a stronger response to the target that's being tracked, but we get a measurable response to the target that they're supposedly trying to ignore. And in this case of vertical vergence, the thing to note is that the functions for the ignored and attended target have pretty much the same shape. It's just a kind of a scaled version that it has a higher peak, but it pretty much starts and ends at more or less the same time. And the second subject is a bit noisier, but the same basic result. So now we have horizontal vergence, and here the pattern is quite a bit different. We still have the strongest response to the attended target, so the red, the inner here, and the green, the outer. But what's different here is that for the case of horizontal vergence, we have this long kind of shoulder on the attended target that is still above chance out at 600 milliseconds. So this we basically think is the result of the voluntary component, that that this quick unattended response is a pure reflex, but that when you're voluntarily trying to track it, that's seen out here at these longer delays. And second subject, same thing. And then here is vertical version. And here it's interesting that there are a lot of oscillations on this response. And we remember that for the cases we've looked at before, the vertical signal didn't have the negative undershoot. But here we see in the unattended response it does. So we basically, this big shoulder that's present for the attended response is gone in the unattended response, and now we've got a negative undershoot. And second subject, same thing. And then finally the horizontal signal. Here it's not quite as pronounced that they're different, but they weaker response to the unattended one and a little bit of a phase shift. So basically these results taken all together have led us to this model of having a reflexive and a voluntary pathway, and that the reflexive system is fast. It doesn't see the second order motion, but it can control all types of eye movements, versus the voluntary system, which is slower but can respond to anything that we can see, but it does not control vertical vergence. So we have this kind of a dissociation of the two. So on the basis of this, we have a few predictions for experiments that we haven't gotten around to doing yet. But one of these is that we think if we were to do a dual ring experiment, but instead of using bright rings like was shown in the demo, if we make rings of texture, so we make the second order rings, that ought to obliterate the response to the unattended ring, according to our theory. And possibly the same thing for a cyclopean stimulus, that is one which is defined by binocular correlation. So I'm not sure how I'm doing for time here. I see it's 25 after, but I know I started a little bit late. I've got another part of the talk that I'm not sure we have time to get through before 2:30, but
Yeah, we have another class starting at 2:30, so you should try to wrap up if possible. And
okay.
then we can take any questions outside. There's going to be some drinks and cookies outside, so
Okay.
there's going to be opportunity to interact there with the speaker afterwards.
Well, in that case, I think I will just make this be my last slide here, and but basically we think these are powerful techniques for learning about early vision, and I'm happy to take questions.
Yeah, we can probably afford a few questions before we wrap up inside here and take it outside. So feel free to go ahead with any questions you might have. Make sure you speak into the microphone if you don't mind.
Yes, it's kind of a real world maybe application of this data. It always struck me that you talked about the luminescence being similar to the background with the circles and so on, that in the real world, a gray car riding along a roadway which is gray seems to be maybe more prone to accidents. Is that something that we might draw from this?
Yes, I think so. I mean, contrast is the number one thing. So
you wonder if insurance companies would take that into effect.
well, the thing is that the backgrounds aren't always the same. So I was reading recently about how they choose what color to paint the military airplanes. Or there's no perfect thing that's good against every background. If you're against a white cloud, you want to be white. If you're against a blue sky, you want to be blue.
Sure. Yeah.
And
I'm thinking there are fewer things out there that are lime colored than are like gray. Roadway gray.
yeah, that's probably true. And I mean, the red sports cars is probably
They get more tickets.
well, but if they get more tickets, it's because they're more visible. And I mean, maybe that's good in the long run.
Do your experiments have any implications for computer vision systems?
Well, I don't know about implications, but the whole problem of how do you the second order motion is kind of an artificial stimulus that doesn't normally, it's not a very natural. Well, let me back it up. That everything usually contains some first order components and some second order components, but it's very difficult in the real world to eliminate all the first order components. So we worry about computationally how you could even process a pure second order stimulus. Is that important for computer vision? Probably not, because it's such an unusual stimulus that doesn't really occur outside the laboratory. You do have things with transparent motion, like where a shadow moves across an object. But again, that's a first order signal that's just superimposed with other first order signals.
Yeah, I want to ask you about how this might apply to early reading, maybe in humans reading text, and the smoothness or lack of smoothness, and what we might learn from your studies, and how to teach reading better, along those lines.
Well, I don't have a simple answer to that, but I will just say that, surprising as it may seem, there is some evidence that that deficits of motion perception are associated with dyslexia, and I don't think anyone has a good idea of why that should be or what, but it's just been observed. And one theory is that, well, maybe when you are moving your eyes across the page, that maybe you have to sort of sense that motion back in order to keep track of where you are. And that if you can't do that very well, then that's why you get these transpositions and get mixed up about what order things came in, is because you're not really matching things up across movements. That's just pure speculation, but we do have hopes that that the basic parallelogram method might be useful diagnostic. In particular, because it measures delays, we think that for diseases like multiple sclerosis, where you have